Now, welcome to the Biocomplexity Institute. I'm representing the Mars Bio Group, and I wish to communicate a little bit of our approaches. To begin with, this is the group itself. So it's a very small group. It's comprised by hopeless mathematicians. Amongst us is Mike Waterman, who keeps us sane if we go too wild once in a while. And we are interesting in identifying structures and structures of any kind. And I will give you a very concrete example. So no worries, I'm not going to bore you with algebraic topology. What I want to showcase to you is how we extract this kind of structures in a more abstract sense, and how this can help in the context of contagion. But structures is probably the important word that shapes the group. And we are pretty much conceptually based in formal methods and information theory. But let me walk you through a pipeline real quick that is very concrete. So in the context of pandemics, we do have multiple sequence alignments. I think everybody in the room has an idea what that is. This means just the method is entirely data-driven, and it has to do with sequences that are aligned. In this case, we're talking about SARS-CoV-2, and the alignment is with respect to some reference sequence that initially basically fathered the whole pandemic. And so we have this. And we ask the following naive question. The question is there particular information in these numbers of sequences that are being overlooked? Fair question. And in order to answer this question, we have to first understand how the data is being processed. And that is, if you have such a collection of sequences, you look at the sequences as individuals, call it a population. And these individuals are understood over time trajectories by creating phylogenies. Now, therein lies a subtlety which already starts in biology with these impossible nomenclatures that are being derived after a subvariant becomes a subvariant, becomes a subvariant, becomes a subvariant. So there are many, many, many very complicated things going on. And then you have a multitude of very, very knowledgeable people agreeing or disagreeing of whether that is a relevant or irrelevant lineage that we are considering and declaring to be of concern. So we take a completely different approach, being ignorant as mathematicians can be, and we essentially say, what if you look at this data set and you're not trying to differentiate it into the rows, but you look at it as a matrix, as a whole. And if you do that, you can think orthogonal. And if you think orthogonal in this matrix, you think about columns. So you start to think vertical. By that, you give up the idea about the individuum, the virus, the piece of sequence that you're actually studying, but you gain something because you look at the whole thing as if it were some kind of superorganism. And when we do this, then we get information about sites, because after all, everything is nicely aligned and it goes back to an original viral sequence that has specific sites. And these are the critical sites that I'm looking at here. And then we look at the evolution of these critical sites. We want to see any type of coordination between these critical sites, irrespective of sequence resolutions. We run standard information analysis, we cluster them. And as we then study these clusters over time, instead of looking at phylogenies, what you traditionally do, we look at the development of such aggregates of sites. We get information about what the virus is doing. I'll show you this in a second. So this is the type of information that you're getting. So you run entirely data-driven a snapshot of the virus for a fixed point in time, and within 3 hours, the algorithm is telling you about functional groups on the genome that you can identify immediately by looking at their co-evolutionary relations. And the idea here is, of course, if the data had structure, I'm the extractor of this structure, of this information. And all I'm saying is that if two sites are very coordinated in their evolution, and there is a certain diversity in that column, then this tells me something. Now, being a mathematician, of course, I can't tell you what it does. But what I can tell you is that this is not random. Now, while this is an example for COVID, you will see that not only can we identify functional units that are also being identified by other methods, we also can actually tell you groupings within functional units. So what you would call a characteristic mutation has a much finer structure that we can identify with this type of analysis. Secondly, and that's going to make everything in physics very happy, is that these systems exhibit some kind of potential. These units that you can identify, the characteristic mutations of potentially bad variants, they behave like particles. And over time, they change. And you can measure these particles as if you were measuring temperature or pressure in a gas. So it's a funny gas, I admit, but information theoretic, it's formally equivalent to having a particle system whose evolution you watch. And whenever this potential spikes, something bad happens in that virus. And we basically did this for Virginia sequence data on a 3-day resolution over long periods of time. And this is the type of output such an analysis is producing. So you see in the upper left, you see that potential. And below, you basically see, I guess, a bar diagram where each of these bars is indicating a particular functional unit on the genome of SARS-CoV-2. And as it persists, basically, we're going to replot it again and again. So many times you see very short units and then they disappear, which is indicative of the virus is trying something new. And once in a while, here in this case, I think it's the first row, you see something persist for a long time. And those are functional units that really matter in the selective process that this virus is being exposed to, right? We see the winners. We see the virus that is capable of infection. And then we can zoom into all sorts of dynamical properties, in this case, some kind of entropy that tells us whether that variant is going to die or it's going to increase its relative percentage within the virus population. Now, this is so sensitive that you can see new variants way below 5% infection rate, way before people are sick in the hospital, way before all that. So you probably see it on the level of 1%, 2%, that you, in a sense, you feel the pulse of that superorganism comprised by many distinct sequences. So what excites Mars biology on about the whole Contagion program is that we will gain an opportunity to interact with different faculties, because although I'm painting a concrete picture and I'm painting a concrete alert mechanism, this obviously is a completely data-driven analysis that is basically an information theory on any kind of data that can be applied in many different contexts, right? Molecular folding being one of them, financial markets are obvious examples, and our favorite is the identification of anything that is artificially introduced into, gene pools, right? Genetically, artificially designed organisms. And so that concludes my presentation. Thank you for your patience.